We have prepared an executive intelligence brief on therapeutic oligonucleotides. Here is a quick summary of our key findings and recommendations. Therapeutic oligonucleotides (TOs) are redefining drug discovery by unlocking access to previously undruggable targets. With unique capabilities to modulate gene expression post-transcriptionally, TOs are at the center of a major biopharmaceutical shift, from protein-centric paradigms to information-directed therapies. Antisense oligonucleotides ASOs, small interfering RNA siRNAs, splice modulators, aptamers, and guide RNAs all fall under the TO umbrella and offer distinct advantages over conventional therapeutics. Despite FDA-approved TOs being less than 20 to date, the clinical and commercial momentum has accelerated sharply. The modality is now transitioning from liver-focused rare disease applications into systemic indications such as cardiovascular disease, CNS disorders, oncology, and fibrosis. This transition is driven by advances in chemistry, delivery technology, and regulatory precedent. TOs leverage Wadson-Crick base pairing to target complementary nucleic acid sequences with high affinity and specificity. By binding to pre-mRNA, mature mRNA, or non-coding RNAs, they can induce gene silencing or modulation without altering the underlying DNA sequence. This targeted approach offers several advantages over traditional therapies. Firstly, TOs enable precision gene silencing or modulation as opposed to inhibition. Secondly, their design and production can be rapid, taking weeks instead of the months required for biologics. Moreover, TOs can target non-coding RNAs, fusion transcripts, and splice variants that were previously inaccessible. Finally, TOs have the potential to be highly scalable with platform features that allow for the development of multiple therapeutic targets from a shared chemistry backbone. However, there are also some key constraints to consider. These include tissue-specific delivery beyond the liver and central nervous system, off-target effects and immune activation, manufacturing cost and scalability for long or modular oligos, and market access and pricing scrutiny for ultra-rare indications. The top 5 strategic players in the therapeutic oligonucleotide space include Alnylam Pharmaceuticals, Ionis Pharmaceuticals, Wave Life Sciences, Sarepta Therapeutics, and Silence Therapeutics. Up-and-comers to watch are Arrow Biotherapeutics, DTX Pharma, Verve Therapeutics, ShapeTX, and Korro Bio. There are 3 strategic implications to consider. First, therapeutic oligonucleotides are vastly expanding the druggable genome. They enable the targeting of intracellular scaffolding proteins, gene fusions, and non-coding RNAs, opening doors to genetically validated but previously inaccessible biology. Second, delivery is the new mechanism of action. With target identification largely de-risked via human genomics, the competitive differentiator will be an understanding of novel conjugates, tissue-specific ligands, and modular delivery vehicles such as peptides, lipids, antibodies, and nanoparticles to unlock extrahepatic tissues such as the heart, lung, kidney, and central nervous system. Third, a platform strategy is a value multiplier. Firms that own a flexible, scalable platform across delivery, chemistry, and regulatory experience can simultaneously pursue multiple indications and partner across therapeutic areas, amplifying capital efficiency and de-risking asset-level failure. In conclusion, therapeutic oligonucleotides are no longer a speculative or niche technology. They represent a programmable, scalable, and increasingly clinically validated class of medicines poised to expand the reach of precision therapy. Future winners will be defined not just by the potency of their molecules, but by the sophistication of their delivery, the breadth of their platforms, and their agility in capturing value across therapeutic verticals. If you found this brief to be insightful, please share it with your network. And if you would like more information, please visit our website. Thank you for watching.